Welcome to another episode from the Veterinary Medicine Digest. In this session, we will be conducting an exhaustive analysis of the architectural integrity of the mammalian axial skeleton, specifically investigating the genetic dysregulations that lead to a rare and debilitating condition known as spondylocostal dysostosis. While this condition has been extensively documented in human clinical literature, its occurrence in canine populations provides a unique opportunity for comparative genomics and a deeper understanding of the fundamental biological processes of somatogenesis. We will center our discussion on a groundbreaking retrospective study involving a litter of mixed-breed dogs that presented with severe spinal malformations, ultimately linked to a novel Splice Acceptor variant in the delta-like canonical notch ligand 3 gene. To understand the clinical significance of these findings, we must first establish the broader landscape of spondylocostal dysostosis. This condition belongs to a heterogeneous group of heritable disorders characterized by congenital malformations of the vertebrae and ribs, often resulting in significant truncal shortening and respiratory compromise. In human populations, the estimated incidence is approximately 1 in every 40,000 births, and the diagnosis is primarily predicated on radiographic evidence of multiple segmentation defects involving at least 10 contiguous vertebrae. These defects often present as hemivertebra, butterfly vertebrae, or block vertebrae, accompanied by a chaotic arrangement of the rib cage, including rib fusions, malalignments, and a reduction in the total number of ribs. A critical diagnostic indicator in young children and infants is the pebble beach sign, where the vertebral bodies appear rounded or ovoid with smooth boundaries, a feature that eventually evolves into irregular ossification as the child ages. The phenotypic spectrum of spondylocostal dysostosis is classified into subtypes based on the specific genetic etiology, with at least 7 distinct genes identified to date in the human genome. These include delta-like canonical notch ligand 3, mesoderm posterior basic helix loop helix transcription factor 2, lunatic fringe, hairy and enhancer of split 7, T-box transcription factor 6, protein Ripley 2, and most recently, delta-like canonical notch ligand 1. All of these genes are integral components or interactors of the notch signaling pathway, which is the primary molecular engine driving the periodic segmentation of the embryonic body into somites. This developmental process is governed by a remarkable biological mechanism known as the somatogenesis molecular clock. During early embryogenesis, the paraxial mesoderm, located on either side of the neural tube, is systematically partitioned into transient segmental structures called somites. These somites are the precursors to the vertebrae, ribs, and skeletal muscles of the back. The timing of this segmentation is controlled by a cell-autonomous molecular oscillator that involves waves of gene expression sweeping from the posterior to the anterior end of the presomitic mesoderm. This clock and wavefront model explains how temporal precision is translated into spatial patterns. The wavefront is defined by opposing signaling gradients, with high levels of retinoic acid in the anterior region and high levels of fibroblast growth factor and Wnt signaling in the posterior region. The intersection of these gradients establishes the determination front, where cells become irreversibly committed to form a new somite boundary. The rhythm of the somatogenesis clock is species-specific, taking approximately 90 minutes in the chicken embryo, 2 hours in the mouse, and 5 hours in humans. These oscillations are maintained by complex negative feedback loops. For instance, the delta-like canonical notch ligand 3 protein acts as a cis-inhibitory ligand, meaning it suppresses notch signaling within the same cell that expresses it. This inhibition is crucial for the fine-tuning of the oscillatory signal required for proper boundary formation. Mutations that disrupt these feedback loops, such as loss of function variants in delta-like canonical notch ligand 3, result in a failure of somite boundaries to form correctly, leading to the severe axial malformations observed in spondylocostal dysostosis. Before we delve into the details of the canine mixed-breed study, it is instructive to look at previous veterinary reports that paved the way for this research. Spondylocostal dysostosis has been reported sporadically in dogs, with historical accounts describing short-spined dogs in Japan and baboon dogs in South Africa. A significant milestone occurred when a frameshift mutation in the hairy and enhancer of split 7 gene was identified as the cause of spondylocostal dysostosis in a family of miniature Schnauzer dogs. This condition was colloquially named the comma defect due to the curved, truncated body shape of the affected stillborn pups. The radiographic phenotype in these Schnauzers was devastating, characterized by sacrococcygelogenesis and severe vertebral and rib fusions. Unlike the surviving dogs in our primary study, these Schnauzer puppies were either stillborn or died shortly after birth, likely due to a total collapse of respiratory function. The genetic mapping techniques used in these canine studies often involve a combination of genome-wide association studies and whole genome sequencing. For example, in a study of Chihuahuas affected by bilateral anterior amelia, a condition characterized by the congenital absence of thoracic limbs, researchers identified a significant association on chromosome 13, delineating a region containing the R-spondin 2 gene. Although this condition primarily affects limb development rather than vertebral segmentation, the research methodology highlights the power of the canine model in identifying mutations within highly conserved developmental pathways. The domestic dog is an exceptional subject for these studies because human-directed breeding practices have created genetically homogeneous populations that facilitate the identification of recessive disease alleles. Now, let us examine the core focus of our discussion, the investigation of a mixed breed litter presenting with varied degrees of Spondylocostal Dysostosis. The subjects were 3 14-week-old puppies, suspected to be littermates, who were presented to a veterinary teaching hospital for a clinical workup. This cohort consisted of 1 phenotypically normal male, 1 affected male, and 1 affected female. The clinical presentation of the 2 severely affected puppies was striking. They exhibited a shortened appearance of the cervical and thoracolumbar vertebral columns and truncated tails, though their heads and limbs were normally proportioned. Neurological examinations revealed reduced range of motion in the neck and tail, but their overall motor functions were surprisingly intact. Radiographic evaluation provided definitive evidence of the skeletal malformations. Puppy 1, which appeared grossly normal, showed only a minor tail truncation with the absence of the caudal end plate of the 6th caudal vertebra and all succeeding segments. Conversely, puppies 2 and 3 displayed a reduced number of vertebral bodies and a multitude of malformations including hemivertebrae, butterfly vertebrae, and block vertebrae throughout the spine. Their rib heads were fused, and they exhibited scoliosis and kyphosis. Furthermore, these 2 puppies showed evidence of a skeletal dysplasia characterized by disproportionate limb shortening, specifically in the humerus and femur, which is a feature not typically associated with human spondylocostal dysostosis, but observed in this canine cohort. To uncover the genetic cause, researchers performed short-read whole genome sequencing on all 3 puppies and compared them to a database of 173 control dogs from various breeds. Because the puppies were found together, their breed heritage was unknown, and researchers utilized commercial breed identification testing. The results revealed that the puppies were full siblings, predominantly composed of German Shepherd dog and Australian cattle dog ancestry. Their coefficients of inbreeding were remarkably high, ranging from 20 to 27%, compared to the 3% average for mixed-breed dogs, suggesting a recent and intense inbreeding event. The variant filtering strategy was performed under a recessive inheritance model, focusing on protein coding variants that were shared by the severely affected cases but absent in the control population. This process initially identified 186 variants, but sequential filtering against the Dog10K database, which contains genomic data from nearly 2,000 phenotypically normal canids, reduced the number of candidates to only 9. After prioritizing these based on predicted functional impact, 4 variants remained on chromosome 1, with a novel Splice Acceptor variant in the delta-like canonical notch ligand 3 gene emerging as the most significant candidate. The variant, identified as a substitution of adenine with cytosine at the -2 position of the Splice Acceptor site preceding exon 5, received a high deleterious prediction score. This highly conserved nucleotide is essential for proper spliciosome recognition. In silico analysis predicted that this mutation would lead to aberrant splicing, potentially causing the skipping of exon 5 entirely, or the use of a weak cryptic splice site. Since exon 5 contains 215 nucleotides, skipping it would introduce a frameshift mutation and a premature termination codon, resulting in an early truncation of the protein. The delta-like canonical notch ligand 3 protein is composed of several critical structural domains, including a signal peptide, a delta serrate log 2 domain necessary for receptor binding, and 6 epidermal growth factor-like repeats. The predicted truncation would severely disrupt the first 3 of these repeats. This is biologically devastating because the 2nd and 5th repeats are sites of essential oxygen fucosylation. Previous studies have demonstrated that oxygen fucosylation deficient divisions of this protein are completely non-functional in vivo. The identification of this pathogenic variant in a homozygous state in the 2 severely affected puppies and a heterozygous state in the mildly affected puppy 1 perfectly aligns with an autosomal recessive mode of inheritance. An intriguing aspect of this study was the phenotypic ambiguity of puppy 1. While this dog was heterozygous for the mutation and should have been an asymptomatic carrier according to traditional Mendelian logic, it still exhibited a truncated tail. This raises the possibility of haploinsufficiency, a state where a single functional copy of a gene is insufficient to produce a normal phenotype. In human genetics and mouse models, haploinsufficiency of notch signaling genes has been known to cause variable and incomplete penetrance of tail and spinal defects. For example, approximately 43% of heterozygous hairy and enhancer of split 7 knockout mice exhibit kinked tails. While heterozygous pudgy mice with delta-like canonical notch ligon 3 mutations typically have normal tails, the Splice Acceptor variant in these dogs might represent an allele-specific effect that manifests even in carriers. The researchers also considered the presence of other genetic modifiers. Within the same 9 megabase haplotype on chromosome 1, they identified a missense mutation in the G-patch domain containing one gene. This gene encodes a component of the spliceosome that promotes splicing fidelity. The authors hypothesized that if this G-patch variant were not neutral, it could theoretically exacerbate the primary splicing defect caused by the delta-like canonical notch ligon 3 mutation, reducing the cellular tolerance for splicing errors. However, based on current alpha missense scores and a lack of established human phenotypes, it was concluded that this variant is likely a neutral polymorphism that co-segregated with the primary mutation due to its physical proximity on the chromosome. Comparing these canine cases to human spondylocostal dysostosis type 1 reveals a remarkable conservation of clinical features. Both species exhibit a short trunk, short neck, scoliosis, and the characteristic radiological patterns of hemivertebra and rib fusions. The survival of these mixed-breed puppies into adolescence contrasts sharply with the lethal phenotype seen in the hairy and enhancer of split 7 mutant Schnauzers, mirroring the human condition where most patients live into adulthood despite their malformations. The phenotypic variability observed between the two homozygous puppies in this litter, specifically the differences in stature and severity of scoliosis, that even with identical mutations, other factors such as environmental influences or subtle background genetic variations can significantly modulate the disease outcome. The environmental component of spondylocostal dysostosis is an area of active research. Studies in animal models have shown that gestational hypoxia can act synergistically with genetic predispositions to worsen the severity of vertebral defects. This effect is often mediated by the downregulation of fibroblast growth factor signaling in the presomitic mesoderm. Additionally, maternal health factors such as diabetes have been identified as risk factors for similar congenital malformations, potentially by disrupting the glycolytic activity gradients that are essential for somatogenesis. The complexity of these interactions is further illustrated by studies of monozygotic twins, where one sibling may present with severe spondylocostal dysostosis while the other remains unaffected, highlighting the profound impact of the embryonic microenvironment. The practical implications of identifying this canine variant are significant for breeding management. Although the mutation appears to be rare, as it was not detected in wider screening of 105 purebred German Shepherd dogs or 76 Australian cattle dogs, its presence in a mixed-breed litter indicates that the allele could be circulating at low frequencies in diverse populations. Genetic testing can now be utilized by breeders to identify carriers and prevent the unintentional production of affected offspring, thereby avoiding the significant animal welfare burden associated with this condition. This is particularly relevant in pedigree dogs where recent inbreeding events can rapidly increase the frequency of rare recessive disorders. Beyond its veterinary utility, the study of spondylocostal dysostosis in dogs serves as a powerful model for human medicine. Dogs are a superior model to mice in many respects. Their size allows for more accurate in utero imaging through ultrasound and computed tomography, and they share the same home environments and medical care levels as their owners. Furthermore, because these disorders occur naturally in the canine population, researchers do not need to maintain artificial laboratory colonies, and the knowledge gained benefits both human clinical research and veterinary welfare simultaneously. As we conclude this deep dive, it is clear that the novel Splice Acceptor variant in the delta-like canonical notch ligand 3 gene is the definitive cause of spondylocostal dysostosis in this mixed breed litter. The findings reinforce the indispensable role of the notch signaling pathway in the temporal and spatial regulation of the mammalian body axis. The convergence of conservation analysis, predicted functional null effects, and radiographic concordance across species highlights the enduring value of comparative genomics in unraveling the mysteries of developmental biology. This research not only expands the known genetic basis of canine skeletal disorders but also refines our understanding of the genotype to phenotype correlations that define rare developmental conditions in all mammals. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging.